So, for decades, we know that light microscopy has been basically our window into the microscopic world. But as we as researchers begin to ask questions about how cells behave within their native environment, within tissues and organs, and then once we realized that, we quickly ran out of the solutions that we could get in standard light microscopy to see deeper structures within the living organism or a tissue. So today we'll look at how multiphoton microscopy has helped us to push past these boundaries to image deeper, clearer, and with less photo damage than before. So firstly, I would like to go over why does deep tissue imaging and multiphoton microscopy that enables deep tissue imaging really matter. Okay. So that is because deep tissue imaging essentially bridges the gap between standard cell biology and how these cells are essentially functioning within their native environment inside of an intact tissue rather than observing these cells in a culture dish. And that is a cornerstone of biomedical research. And what deep tissue imaging does here for us is that it enables visualization of biological structures hundreds of micrometers to few millimeters below the surface of living tissues, intact tissues. And what we can do is we can capture high resolution 3D images without the need for extensive sectioning with a microtome or without extensive photo damage. And what that enables us to do is to study complex cell-to-cell interactions and how these cells interact with each other and how cell-cell interactions can influence tissue development and organ development. And we can observe this within the native environment of the cells. This also enables real-time studies of, like I said, developmental biology, or for example, study of neurons and also immunology. And using this, we can essentially, for example, if we are studying a gene, and if we mutate that genes, how cells interact with each other, what defects do we see in that, and how this translates into the broader developmental defects within a tissue or an organ. That is what we can study with a multiphoton microscope. So even though the most of the usage of the multiphoton system is restricted to life sciences, it is now emerging as a potential diagnostic tool in clinical research. So for the moment, it is mostly used for studying, for example, spheroids, where drug studies can be done. And then since spheroids are really large structures, standard confocal microscopy, as I'll explain later on in the next few slides, is not enabling us to image deeper. Okay. So that's one thing that can be studied in terms of preclinical research. And also we can look at, for example, cancer tissue or any patient tissue and stain it with certain antibodies and dyes and uh, look and study with deeper imaging what kind of disease or abnormalities or how these diseases and abnormalities can alter the tissue structure. So even though the topic is multiphoton microscopy, I am aware that not all of the attendees in the meeting might be having an up-to-date background in fluorescence and confocal microscopy. So to make sure that once we reach the step of multiphoton microscopy, we are all on the same page. I'd like to start off with a bit of basics. So before we dive deep, it's also important to study the broader evolution of imaging. So I'll start by explaining basic contrast methods that are used in bright field and phase contrast microscopy. Then afterwards, we move into fluorescence-based techniques, which also forms the bedrock of multiphoton microscopy. And then look at how fluorescence microscopy and then advanced confocal microscopy may not be fully suitable for deep tissue imaging. Yeah. So the drawbacks of these two techniques. And then finally, what are the core optical components of a multiphoton microscope? What is this, roughly, what is the optical setup of a multiphoton microscope? I'll explain grossly. And then what are the advantages of multiphoton microscopy, which we are not able to achieve in standard fluorescence and confocal microscopy. And then lastly, I'll be explaining what are the recent advances in terms of optics that are used in deep tissue imaging for multiphoton microscopy. So this presentation overall will be a conceptual overview of the technique. And if any of you would be having any specific application questions of your own projects or of your own experiments, or questions about a complex optical setup that you might be looking to assemble in your lab. We can come to that in the Q&A session afterwards. So coming to the basics of a microscope. So at its core, a microscope serves two main functions: to make tiny structural details visible, and then equally important is contrast, because most biological specimens are completely transparent when illuminated with visible light. So we need magnification and we need contrast. So how to increase contrast? So to increase contrast, what we have to do is, one way to do is chemical staining. So for example, you might have seen such kind of images for hematoxylin and eosin staining. So in H&E staining, we can label certain cell components like nuclei or some components in cytoplasm with some stains. And then that generates contrast that enables us to look at different parts of the tissue or different parts of the cell. The other way to do is to exploit differences in refractive indices within a sample. So for example, there is a difference in density between the aqueous cytoplasm and the cell nucleus or other such organelles. And what that does is that when light travels through the cell, it travels slower in the regions with higher refractive index. Okay. And these regions tend to retard or bend the light, which produces phase shifts. And that is what bright field microscopy essentially exploits. So white light passing through the sample is then modulated by different organelles and different structures in the cell. And then this contrast that arises from this certain absorption scattering is then amplified in a bright field microscope. So if this is our sample over here, and that is a light passing through. So different kind of structures in the cell will interact with light differently. And we, we also have undifracted light over here, and then we also have scattered and diffracted light. And the interference pattern that this creates on the image plane is what we see as a microscopy image. What are the disadvantages of this particular bright field microscopy? So since we are now talking about microscopy, just bright field, where we are not staining or labeling with any dye or so. So in this case, the internal structures are very poorly visible. So we are not able to, for example, look at different organelles like mitochondria or endosomes or so. Or the nucleus may be made out up to some extent, but not other organelles. And then, like I said, it's difficult to gain any structural insights without using dyes or stains. So one more technique that was invented to essentially overcome these limitations was phase contrast microscopy. So in phase contrast microscopy, what essentially is done is that proximal to the condenser, a phase plate is introduced, and then an annular phase mask is introduced in the objective. And what it basically does is it modulates the light path that was in the bright field, the light path was like this. And here the light path is essentially modified. Okay. And this, what you see here, is unscattered light path. So when we insert a sample in this particular field path of light, we see that this sample is, because of its internal organelles and internal structures, it is scattering light. And then this scattered light, just a second, I'll just move this. Yeah, and then you can see that this phase ring that is over here, it is manufactured such that the undiffracted or unscattered light is passing directly through like this, and then the scattered or diffracted light is essentially being retarded. And what this essentially does is that it is enhancing the contrast that you saw in the earlier bright field, and you see even better contrast with phase contrast microscope. So this is how we can see the evolution of contrasting techniques. This is the bright field. So this is the same cell image with bright field microscopy and phase contrast. So here you can see that the cell, since it's transparent, you're less able to make out what is inside the cell, what are the different organelles there. You can make out the nucleus, but not other things. In phase contrast, you have slightly better idea of what could be happening inside the cell. For example, if you want to look at mitosis, you might be able to see the chromosomes that are being separated during mitosis. And then another example here that shows the same thing. So the major disadvantage of these two techniques is that even though phase contrast is giving us slightly more information of the content inside the cell, it is essentially not telling us specific information. So for example, if I have to look at a specific protein inside a cell, and if I have to look at a specific cell inside of a tissue, That information we cannot gain from a phase contrast image. So there we need to turn to fluorescence microscopy. Okay. So fluorescence microscopy essentially allows us, like I said, to tag specific proteins using dyes or antibodies. And then these proteins, what we can call as light up, and then we can essentially localize these proteins inside a cell, and we can localize specific cells inside a tissue. Yeah, so optical contrast methods like bright field phase contrast or DIC or so show us where cells are, and fluorescence microscopy shows what's particularly inside of them with high specificity. So for example, I have here two images from my PhD days. So here is a Drosophila larval brain. And here you can see that the different kind of labeling I've done. For example, this is a marker of neural stem cells. And I can specifically visualize neural stem cells. This is a marker of dividing cells. I can make out those cells too. And then I can study what is a normal morphology and what happens in case of a mutation. So here I'm labeling specific cells inside of a tissue, and here I have an individual cell imaged here. And in this cell, I can image a particular protein where it localizes. I can image, for example, spindle microtubules with the tubulin with an antibody specific to tubulin dimers. And I can visualize the nucleus also. So specific components can be visualized by using dyes or antibodies, and we can get this precise information. So fluorescence, which is forming the bedrock of both fluorescence microscopy as well as multiphoton microscopy, is essentially taking place when a photon of high energy is hitting an electron. Okay, and then when this photon is hitting an electron, it is essentially acquiring this energy transferred to it by the photon and is moving through energy levels from the ground state to the excited state. So it is absorbing the energy of this photon and moving to the excited state. But since this electron that is now absorbed, this energy is unstable at the excited states, it undergoes non-radiative transition. It slowly steps down the energy levels, and then finally it makes a big jump back down to the ground state. And when it comes back down to the ground state, it emits the energy that it absorbed previously, and this energy is emitted as a photon. But what happens is that the absorption is of a certain energy, right? So the photon is hitting the electron, it's absorbing it and going to the excited states. But since the electron is undergoing this non-radiative transition and coming down some steps, it loses some energy non-radiatively, and therefore when it comes back down to the ground state, the energy that it releases is less than what it absorbed. Okay, so excitation of an electron happens at a higher energy, and then the emission happens at a lower energy. So a lower energy photon is emitted when the electron is coming down to the ground state. So that is the essential principle of fluorescence. But what does fluorescence mean in the context of, for example, what is high energy photon, what is low energy photon, and how really we can see fluorescence through this principle? So this is the electromagnetic spectrum, and you can see that certain ranges of wavelength correspond to certain components of the electromagnetic spectrum. For example, gamma rays, X-rays, UV. And what we are interested in terms of fluorescence microscopy is visible range. Okay. So this visible spectrum roughly corresponds to around 400 to 700 nanometers. Okay. That is the wavelength of visible spectrum roughly. And here we have, for example, at the wavelength of 450 nanometers, we have blue light. At around 510 to 520 nanometers, we have green light. So now going back to the principle of fluorescence. Now, if this electron is absorbing, for example, a photon of wavelength 400 nanometers, for example, a blue light photon, then it goes to the excited states. When it comes back down, it releases a photon that is of energy of around 500 nanometers. So it is absorbing blue light and emitting green light. So that is the concept of fluorescence. So for example, if the dye, the particular dye can be excited with a wavelength of 500 nanometers like green, then it will be excited to the excited state with a green light, it emits red light. So that is that is how we are seeing fluorescence. So we are exciting with one wavelength and the emission is happening in another wavelength. And here, one important formula to keep in mind is that energy is inversely proportional to the wavelength. So higher energy, so for example, higher wavelength has lower energy and lower wavelengths has higher energy. That is the reason why here blue light that is of lower wavelength but higher energy, is used for absorption. And then when the emission, which is of a lower energy, is having higher wavelength. So please keep this in mind because we would need this information to understand how multiphoton microscopy works and what is the exact meaning of multiphoton. And in case you have any questions, please don't hesitate to interrupt. Okay, so how does a fluorescence microscope essentially exploit the excitation emission of a fluorescent molecule? So here we have a light source, a multi-wavelength light source, and then we have different kind of wavelengths getting emitted from this. And here we have an excitation filter. So for example, if we have labeled the cell, or if you are expressing in the cell green fluorescent protein uh, labeled sample. So here the excitation filter will make sure that only the wavelength that is needed to excite the green fluorescent protein is allowed to pass. Then this dichroic mirror will reflect that wavelength onto this objective to the sample, and then the green fluorescence that this sample is emitting is then transmitted through by this dichroic mirror to the ocular to the detector. So that is essentially the principle of a fluorescence microscope and the rough optical components of a fluorescence microscope. And then, like I mentioned before, we can use different kind of fluorophores or different kind of dyes to label certain organelles or certain molecules in the cell. And we can also use antibodies that recognize certain proteins. And then we can tag these antibodies with fluorescent molecules so that we can visualize these structures inside the cell. Okay. So fluorescence microscopy is excellent for thin samples, but the problem arises when we are looking at depth. And the problem here is in wide field microscopy, the entire sample is illuminated. So for example, this is your tissue. Just imagine that this is your tissue. And if you're firing it with a excitation light from a fluorescence light source, it is exciting entire sample. And what that does is that it causes fluorophores above and below the focal plane to be excited. And this creates an out of focus background fluorescence. So whatever plane that you're focusing on, you're getting background fluorescence from all the other planes in the sample because the whole sample is getting excited and its whole sample is giving the emission. Okay. So that is why you get a lot of blur when you are imaging even like a cell monolayer. And then as the imaging depth increases, that is the if the tissue that you're imaging or the structure that you're imaging is deep and thick, then there is even more scattering and absorption, and that causes the excitation and emitted light to degrade contrast and resolution. And furthermore, if you are imaging light samples, since the entire sample is getting illuminated with your excitation light, you have high photobleaching and phototoxicity throughout the sample. And this is why the depth at which you can image with the standard fluorescence microscope is around 50 to 100 nanometers in most biological tissues. So there are certain improvements that were devised to make sure that at least the light that we are collecting is not collected from the entire sample, but from certain optical sections. So this advancement is also known as confocal microscopy, which can do optical sectioning for clearer images. So what confocal microscopy, as many of you would be knowing, essentially does is that it has a pinhole in the light path. And what this pinhole does is that when the emitted light is coming up from the sample through the objective, it is placed such that it is blocking the out of focus light. So the focal plane where you are imaging, light only from there is being captured in the detector, and all the out of focus light is blocked. As a result, the blurriness that you see in the wide field image due to collection of light from all the focal planes is then corrected here. And then for a confocal image, you essentially see image which is very high contrast and sharp as it is collected only from a single focal plane. So we get a sharper high contrast image, and we can also do 3D reconstruction from sequential optical sections. But the problem again is that just like wide field microscopy, here in confocal microscopy, even though we collect light only from one optical section, we are not preventing the illumination of the entire sample. So here also the entire sample is illuminated, and that is why this also results in photobleaching and phototoxicity in the entire sample. And also one disadvantage is that confocal microscopy and the dyes and antibodies which are used for confocal microscopy are tagged with fluorophores that emit visible light in the range of 400 to 700 nanometers. And again, for this reason, there is high scattering and absorption rapidly when you do excitation and emission in a deep tissue. And that is also the reason why imaging depth in this case is limited to 80 to 120 micrometers in most biological specimens. So then how do we achieve this requirement that we essentially want to do deep tissue imaging while preserving tissue integrity? The answer lies in multiphoton excitation. So here what we do is we use longer wavelengths. So longer wavelengths have lower energy. So they scatter less in the tissue. And then we do localized excitation. So for example, in confocal, we illuminate the entire specimen. In multiphoton, we do point illumination. So only the focal plane where we are currently imaging, only that focal plane is then illuminated with the light. And that is why we illuminate one optical section, we collect light only from that optical section. We don't need a pinhole. And also that is the reason why we are not having photo bleaching, because we are only illuminating a single spot, not the entire specimen. So how does this work? Because we have seen earlier that a photon of higher energy is required to excite a certain fluorophore. And then when it emits fluorescence, it emits at a higher wavelength and lower energy. But since the multiphoton is relying on high wavelength light, that is the infrared light, how is it possible that we can use this for imaging? Because most of your samples will be having, like, for example, GFP or any nuclear stain. So all these dyes, stains, and fluorescent proteins are emitting in the visible range of wavelengths. So how can we do excitation with a high wavelength light, which is of a lower energy, and expect to get emission of lower wavelength, which is of high energy? So instead of single photon excitation that is done in standard fluorescence microscopy, like this over here, what we do is we excite with not one but two photons of higher wavelength. So since two photons are being used for excitation, it is still sufficient to push the electron to the excited state, and then we can get the emission in the visible spectrum. So the core principle is we use two photons of higher wavelength, that is the NIR wavelength, to excite the sample, and then the emission that we are seeing is in visible wavelength of high energy. Then typically the NIR light that is used in multiphoton is in the range of 700 to 1100 nanometers. That penetrates deeper into biological tissue, and this enables deep tissue imaging of up to 1 millimeter in some cases, and provides optical sectioning because we are only illuminating the focal plane where we are imaging. So what are the optical components, and roughly what is the setup of a multiphoton microscope? So firstly, what we use is a mode-locked pulsed infrared laser with high peak power. So this is a continuous laser that in the confocal microscope that you might have used, you have essentially a continuous laser. But in a pulse laser, what's happening is that it is emitting light not continuously, but in pulses. And the duration of each pulse is roughly, in many multiphoton, you can alter this, but in most cases, it is around 100 femtoseconds. And why is it 100 femtoseconds? Because in that duration, you can very efficiently send two photons to excite your sample. And because of these pulses, since this two photon excitation can also result in very high heating of the sample, because it's IR light, and then the energy that you achieve, the peak power that you achieve at your focal point is around 100 kilowatt. So that can cause heating, and that can also damage your sample. So that is why this light is not sent continuously, but in pulses. Yeah. And then you can modulate the frequency of these pulses to make sure that you can do fast imaging. So for very fast imaging, you can keep the frequency very high. And then for slow localized imaging with high emission intensity, you can keep the frequency lower in the kilohertz range. So that's how it's done. We also use objective lenses that are able to focus the NIR light tightly onto the focal plane of the specimen. And then this is using this, we can achieve high contrast and resolution within deep tissues, and we can avoid scattering. And what we also need as an optical component over here, so apart from the light source, we also need here scanning mirrors. So in confocal microscopy also, the scanning mirrors are used. So these scanning mirrors are focusing light onto the sample here, and there is a movement between the two mirrors. There is a raster scanning using these two mirrors, so you can scan your sample using the scanning mirrors. You can also use something called resonant scanner to increase the speed of scanning. Yeah. And what we also use here are something called non-descan detectors. So in normal confocal microscope, what will happen is that your excitation light will be focused onto the specimen using these mirrors. And then the emitted light will travel back through the same path to the detector. So here what will happen is that for a deep tissue imaging, this is not very ideal is because you're collecting photons from very deep inside the tissue. And often this gets so deeper and rarer photons from deeper in the sections get diluted in this process, and then the collection is not very efficient. And that is why in multiphoton microscope, the emitted light is not going back through the same path like in a confocal microscope, but rather it is being collected in a non-descan detector, which is which is very close to the objective. And then this enables that you collect the highest possible photons and increase the signal. And it's a rough comparison here between confocal microscopy and multiphoton microscopy. And then here the excitation light, as I mentioned, is in a visible range at 400 to 700 nanometers. And with multiphoton, we have excitation with near infrared light in the wavelength range of 700 to 1100. So the excitation is being done in confocal microscopy along the entire beam path. So the whole sample is illuminated. And multiphoton microscopy, illumination is happening only at a focal point. So for optical sectioning, if you want to generate images from single optical section, here to reconstruct it into 3D format. Here for optical sectioning in confocal, you need a pinhole, and in multiphoton microscopy, it is intrinsic. Since we are only exciting the focal point, we are only collecting signal from the focal point. So we are not needing to minimize any out of focus signal. And because we use near infrared light for excitation, it scatters less and we can go deeper into the tissue and we can go up to 1 millimeter also. And the imaging depth, what we can achieve with the confocal is around 100 to 120 nanometers. And since confocal microscopy illuminates the whole sample, we have significant photo bleaching and damage when we try to do long-term deep tissue imaging. And here it's not the case because excitation is limited to the focal point. And signal detection, like I explained in the previous slide, for confocal microscopy, the emitted light is coming through the same path as the excitation light, and it is going back through the pinhole. And in the multiphoton microscopy, the non-descanned light, which is non-descanned detectors, which are placed close to the objective, enable efficient capture of the photons. And that's why you'll get a better contrast and better signal. And the special modes here in multiphoton, so in confocal, you can only look at fluorescence imaging, but in multiphoton microscoping, apart from fluorescence, there are specialized imaging techniques, for example, second harmonic generation imaging, which is label-free imaging. I'll be explaining that very briefly in the next few slides. So that were the major differences between a fluorescence and confocal microscope and a multiphoton microscope. And coming to the broad range of applications that we can put the multiphoton microscopy to use, we essentially can use it to in neurosciences to look at in an organism or in a, for example, a zebrafish tissue or in Drosophila brain, for example, how the neurons are how the neuronal axons are innervating the tissue. And if we want to track, for example, calcium imaging or so, that can be done. In context of cancer biology, we can look at large spheroids or organoids, which are otherwise not possible to image through standard confocal microscopy. And if we have, for example, a mouse where we have injected cancer cells and we have to visualize a tumor and a tumor microenvironment and angiogenesis and such kind of applications, we can directly mount this xenografted mouse onto the stage of the microscope. And then using multiphoton microscopy, we can look at the cell-cell interactions and process such as angiogenesis and cell migration within the living animal in the tumor tissue. For developmental biology as well, we can use the multiphoton microscopy to image, for example, the development of embryos with minimal phototoxicity. And for translational clinical researches, multiphoton is still not widely used, but then in future, it can be used for label-free imaging of skin and cornea and other tissues, for example, for early disease diagnosis. So this is one example that we have acquired on our multiphoton system. One of my senior application managers has acquired this. So this is a 10-day post-fertilization zebrafish. And here here this zebrafish is a transgenic zebrafish, and it is expressing a certain protein that localizes only to one subset of neurons. And you can see how beautiful this image is in terms of the depth. And then here in this image where I have shown the 3D reconstruction, we were able to go down to a depth of 400 nanometer, 400 micrometers. And with confocal microscopy, if we do the same kind of imaging, we'll not be able to go deeper than, for example, 80 to 100 micrometers. So that is the kind of image quality that you get with a multiphoton microscope, and then that's how it compares with a confocal in real world samples. And then one more application is something called a second harmonic generation imaging. So second harmonic generation, so the basic principle is that two photons of the same wavelength are essentially interacting simultaneously with certain protein polymers with repeating structural units. Okay, so for example, if you have a collagen, if you have microtubules and myosin that have repeating structural units, in this case, what happens is that these photons, like classical fluorescence, these are not absorbed, but instead they are combined into a single photon of half the wavelength, that is double the energy. So the excitation that you're doing is with NIR light, and the emission that is coming is visible light of higher energy. And this is process is different from fluorescence because there is no absorption and emission, but rather these photons are combined because of the structural properties of the sample. And that's why we see fluorescence. And but this is only visualized in certain biomolecules. For example, the collagen that is labeled collagen that is visualized here is visualized through second harmonic generation. So collagen is labeled in gray, and the green is in another protein. So this visualization was done without any kind of labeling, and just with second harmonic generation, we were able to visualize the collagen. And for this reason, it is label free. No dyes or stains are required for these kind of biomolecules. It causes minimal photo damage because there is no absorption. There is only scattering, and we can get the information about the structural organization, alignment, and orientation of such kind of macromolecules. So these were some of the examples and of where multiphoton microscopy can be used. So we now see that how it has revolutionized deep tissue imaging, but the story of multiphoton microscopy does not end over here. So a number of exciting developments are now pushing its capabilities even further. So starting with three-photon microscopy, three-photon microscopy is using even longer excitation wavelengths, typically around 1300 to 1700 nanometers. Okay, so these photons scatter less in tissue, allowing imaging at depths beyond 1 millimeter. Okay, for example, through the full cortical thickness of a mouse brain, right? You can use this microscopy technique. And then three-photon process also confines excitation even to a smaller volume, even to a smaller region or smaller focal volume, and therefore gives exceptionally clean optical section. Then we have adaptive optics. So this is another, like for example, an enhancement to existing multiphoton microscopy. And here what we do is these adaptive optics compensate for distortions in the light wavefront that are caused by the different refractive index of the different compartments in the tissue. So by using specialized mirrors and optics and spatial light modulators, we can correct for those aberrations, and this improves resolution and signal quality when doing deep tissue imaging. Then we move to the miniaturized and green lenses. So green lens is essentially a gradient index lens, or it is some kind of a micro endoscope that is inserted very close to the animal where you're imaging, and this enables deep tissue real-time imaging of neuronal activity or other biological processes. And again, this can be applied to whole animal imaging. So, of course, and then since with the advancement of all these optical components, we also have advanced detectors and tunable lasers. So, modern femtosecond lasers are more tunable and stable, allowing excitation of multiple fluorophores, or even allowing three-photon excitation. At the same time, detectors, for example, sensitive gallium arsenide phosphide and other hybrid detectors are also coming up to improve photon collection efficiency, which is very crucial for deep tissue imaging where the signals are weak. And then, of course, we have label-free non-linear modalities like second harmonic generation. And then techniques such as CARS, for example, This technique gives chemically specific imaging of molecules such as lipids and all complementing fluorescence-based imaging. So together, all these advances are expanding the frontiers of optical microscopy, allowing us to image deeper with more detail and with less disturbance to the living system. In summary, so multiphoton microscopy has revolutionized deep tissue imaging, and these ongoing advances that I explained before are further pushing the boundaries that we have already achieved with multiphoton. And therefore, whenever you think of deep tissue imaging or whole animal imaging and imaging of interactions of precise cell-to-cell interactions deep inside living intact tissues, the go-to solution that we have here is multiphoton microscopy. So that brings me to the end of my presentation. So I thank you all for your attention. I hope this talk gave you a deeper appreciation of how these various advances in multiphoton microscopy and the applications to which this technique can be put to use will transform our capacity to visualize complex biological systems. So thank you for your attention. And I'll also like to let you know that if you would like to know anything more about these kind of optical microscopy solutions, kindly write to us at enquiry@dssimage.com, or also you can write to me personally. And again, once again, thank you for attending this webinar, and thank you for your attention. And if you have any questions, I would be happy to discuss them. Thank you.
Thanks a lot, Dr. Rohan, for the wonderful session. I believe everyone had a learning. So, this is to request to all the participants that we are open for the Q&A session. So, if you have any question, you may unmute yourself and ask the question, or you may even drop in the chat box, whichever is more convenient to you. So, I see that in the chat box there is a message dropped by Vishal Singh. He's saying to please share the recorded PPT after the meeting. Yes, we will share the recordings in a day or two, basically, and we'll email you all the recordings. So we are open for the Q&A.
May I ask one question, Mr. Rohan?
Yes.
I'm still confused after seeing all your photon and confocal things. See, as a
Yes.
pathologist, I used to attend stent slides, and
Yes.
I feel very comfortable seeing tissue structures under bright microscope. Okay, of course, the slide is very thin and
Right.
stay in there, then only we can watch. Now, is there anything parallel to this? Because if you say this multiphoton microscopy, of course, it is very expensive also. And then what
Yes.
we see there, only light reflected or light absorbed tissue structure. So I am still not convinced whether that kind of contrast is visible in the latest microscopy.
Yeah, so the advantage also is that just like in confocal, you can label specific structures with antibodies. So, and then the antibody labeling will give you that required specificity in your sample. And that specificity is not utilized fully with a confocal because of the scattering. It only goes to a certain amount of depth, like 50 to 100 micrometers. But if you want to visualize in a large organoid or so, or in a large tissue, without like physically sectioning it with a microtome, then in that case, multiphoton microscopy will give you an advantage that you can process it just like you do for a fluorescence microscopy. You don't need to physically section it, and you can go as deep as 1 millimeter.
Only depth we can attain. Depth of
Yes.
the tissue, right? Otherwise, fluorescent, as you said, we can tag the antibody. We can use immunohistochemistry also after tagging, using chromosomes different.
Yes, but if we use chromogens, exactly as you said, we can use chromogens, but then you might need to rely on bright field microscopy or phase contrast. And there, if you want to do optical sectioning, the ability of optical sectioning will be limited. So if you want to, for example, if the tissue is a few millimeters thick, and if you do an staining with hematoxylin eosin, for example, then you would be having overlapping layers of the structures that you want to see in a tissue. And then if you want to section, for example, as a small section, so in a multiphoton microscope, for example, when you do a Z stacking to image in 3D, the sections can be as thin as 0.01 micrometers. So that kind of resolution you can get with a multiphoton or with a confocal microscope. So that you'll not be able to achieve with a color microscopy with hematocyte labeling, for example.
That is the only advantage, that multi-layer tissues we can examine at one place using
Yes.
fluorescent dye.
Yes.
What about this 3-photon and 2-photon? As you told, we don't label free visualize.
Yes.
How much structure we can see level free visualize?
So, yeah. So, the structure, if you compare, for example, collagen labeling with an antibody to the label-free imaging, the antibody labeling or labeling with a dye to collagen will give a lot better contrast and lot better specificity. Here in label free, we don't get that kind of specificity, but this is essentially useful example. For example, if you're imaging a live tissue and you are not able to tag it with a fluorescent dye or with a fluorescent protein, and then you're comparing, for example, a disease condition, for example, a skin disease condition where collagen structure can be altered as compared to normal condition. So in such cases, this label-free imaging can give a specific advantage. It will not be as specific as labeling with antibodies, but then to compare a disease condition versus a normal condition, when you are not able to tag it with any fluorescent dye, this will give you an advantage.
Yes, that's what actually I wanted to know. Because as you said, collagen we can see, but suppose there are, I'm cutting an artery and I'm seeing different layers, collagen, laminate, and maybe certain endothelial cell layer. Those three layers, can I differentiate by level three? Uh, right? No.
So I think your voice was getting cut. Sorry. Could you please
So I'm
repeat the
saying,
question? Sorry.
suppose I am
Yeah.
watching a longitudinal section of an artery, which has
Yeah.
by endothelial layer, then maybe connective tissue layer which contains
Yeah.
collagen or laminin. These are different protein structures.
Yes.
So without dye, is it
Yes.
possible to differentiate
Yes.
them
Yes.
to level three? No. Or yes, I don't know. You have to tell.
Yeah. So yeah. So that's a very good question, actually. So the real strength of this second harmonic generation imaging is that, for example, like you mentioned, that you have to differentiate between laminin, and other kind of different extracellular matrix components with the deep tissue imaging. So here you can combine, you can't visualize apart from collagen, microtubules, and myosin. There are not many other tissues that you can directly visualize with the second harmonic generation. So the second harmonic generation can be used, like it can be paired with a fluorescent labeling. So for example, if you have GFP or M-cherry labeled laminin, for example, and then you have collagen with a second harmonic generation. So you can visualize the different localization patterns or the morphologies using this approach.
Thank you. Thank you.
Yeah.
Very nice presentation. Congratulations.
Thank you, sir.
Dr. Rohan, so there are a few questions in the chat box. Are you able to see them?
Okay, I will try.
If not, let me know. Just give it a try.
Yes.
Yeah.
So, Yogaraj ji, so you have asked a question about how to selection the excitation wavelength in multiphoton having 800 nanometer to excite protein labeled with 488 nanometer, 546, etc. Very good question, sir. So, in order to do this, as I mentioned, we have to send 2 photons of high wavelength, low energy to get excitation of high energy and low wavelength. So, what essentially we do in a multiphoton microscope is that people have optimized what kind of wavelength that you need to use, to excite, for example, GFP or to do excitation of any green dye. So, we normally use a wavelength of 920 nanometers of a multiphoton laser to excite GFP, for example, which has a emission at 488, or any kind of green dye like FITC or hexafluor 488. So for DAPI, which is a blue emitting dye, we do the excitation with 720 nanometers. And for a green dye, we do excitation with 920 nanometers. For Alexa for 546 or for M cherry or so, we do excitation in the range of 1020 nanometers to 1040 nanometers, 1040 nanometers. So that's how we do the excitation. And then we have one more question. Is there any method from Shrikant ji?
Sir,
From
sorry,
Shrikant
sorry
Sridhar?
to interrupt you, sir. Can I
Yeah, sure,
explore
sure,
anything?
sure, 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 sure.
Yeah, thank you for nice
Thank you.
presentation. Actually, I am using multiphoton photon microscope, na? Parietal
Okay,
microscope. It has only 800 nanometer that laser.
okay, so okay, so it is not tunable. It has a fixed wavelength.
I, it is not tunable. It is very old system by Olympus,
Okay, okay,
almost 10 years past.
right, right, right.
Then
So
in such case, what can I do?
yeah, so in case that your laser is mode locked to only give excitation at 800 nanometers, I would
Yes,
suggest
sir.
you to use dyes that are more blue shifted. For example, if it is possible
Okay,
in your condition to avoid using M cherry, for example, and if you can continue to use, for example, GFP or CFP, for example, or YFP. So such kind of proteins, if you use, you can achieve efficient excitation of these fluorophores with an 800 nanometer laser. So it is also possible with the same laser to excite a blue dye and a green dye and a green fluorophore like GFP. It is possible. But since it is mode locked at 800 nanometers, the emission that you'll get out from it will be not as efficient as what you can achieve at 920 nanometers. But I would suggest you to use as much of blue shifted proteins or fluorescent proteins
okay,
or blue shifted dyes as possible.
okay. Thank you so much for
Thank you.
clarification.
Thank you. So, if we can go to the next question. So we have Shrikant ji here. So Shrikant sir. So he's asking, is there any method to reconstruct the 3D image of the microscopy? So returns of the cells and tissue interaction. Yes, definitely. So, so in our case, we are essentially working with the Olympus multiphoton microscope. So here we are providing a software called Cell Sense Dimension packaged together with the Olympus microscope. So here, by default, it contains a software for 3D reconstruction. So in that you can visualize different layers of your tissue. You can try to locate your specific cells of interest and see what other cells are coming in contact with these cells. So with different kind of, if you use 2 to 3 fluorophores to label your specific cells and a marker to label the tissue, you can essentially do 3D reconstruction and you can do this kind of specific identification of cell within different layers of the tissue. Yes, it is possible. Yeah. Thank you, sir. And then we have another question from Shantanu ji, Shantanu Mandal. And the question is, which type of crystals are used to generate this single harmonic generation and third harmonic generation for multiphoton microscopy? So I'm not sure by crystals, are you referring to what kind of material the multiphoton laser is using? Because here what we are doing is that we are not crystallizing any proteins here. So the proteins are there within the living tissue or the intact tissue. And because of the structural properties of the protein, we are essentially getting this second harmonic and third harmonic generation. Yeah, so we are not crystallizing the proteins. So I'm not sure if I answered your question. Feel free to also come in for more clarification. So any other questions from the audience?
So I think that's all it is.
So if anything else you might be wanting to ask, please feel free to contact us, as I'll again briefly share the contact slide. So here are the contact details of the company. So please send any, for example, Yogaraj ji, you are mentioning that you have only a single mode locked 800 nanometer laser. So in future, if you would potentially like to upgrade this system to have a multi-mode laser which can image at different wavelengths, so please feel free to reach out to us at enquiry@dssimage.com, or you can also write to me at rohan.chippalkatti@dssimage.com, and we will be more than happy to help you.